too inundated with different names of different organisms and how they cause diarrhea because the story is about to get much more complicated. Today, we are going to be discussing parasitic agents of diarrhea diseases. By the end of today's discussion, should be able to discuss some of the parasitic infections that present with diarrhea or dysentery. You already know what dysentery means. You should be able to discuss the etiology, organisms are transmitted to a susceptible host, the life cycle, since we're discussing parasites. Remember that a very important component involving a parasite includes the life cycle, the pathogenesis, laboratory diagnosis, and how infection can be prevented. You still remember this table I showed you in the very first lecture. Remember I told you that we have a diverse agent that could lead to GI infection. So we're going to focus on protozoan parasites and helminths today. We start off with the protozoan parasite Giardia lamblia, which causes the disease. It is a protozoan parasite and it has a predilection for the gastrointestinal tract. It is a flagellate, so that means it moves with the aid of flagella. The common reservoir of this agent include humans, particularly asymptomatic patients who are shedding the infectious any clinical symptoms. Wild animals such as beavers will be infected without any symptoms, so they will continue to deposit the infective, including contamination of surface waters. Domestic animals such as dogs may also be infected and they could serve as reservoirs. And then for people who go on camping trips, hiking and what have you, coming in contact with contaminated stream water could also be a risk for infection because this stream could be a reservoir. How is the infection acquired and who are the likely people to be infected? Fecally contaminated water, because the cysts that are infective are usually passed out in stools. So ingesting stool, as in ingesting it literally, is the mode of transmission. So fecally contaminated water or food can transmit the infective cyst. Then for baby handlers working with infants, working with young children, you may have hand-to-mouth transfer of the cyst. This could occur when you have these minders changing diapers, when you have a patient who is not symptomatic but producing the cyst, and going on ahead without washing her hands to feed another baby. So you can have transmission in daycare centers. Infection can also be readily spread amongst family members. All it takes is for one member of the family to come down with infection or to be asymptomatically infected, and other members of the family are at risk. It is also transmitted through oral nasal sexual practices. So in homosexuals and in people who have peculiar sexual orientations, this agent can also be transmitted. The organism is also referred to as Giardia intestinalis because it's found in the small intestine, or Giardia duodenalis to indicate that the duodenum is usually the site that this organism attaches to. It exists in two forms, as you can recall from your PhD lectures, the cyst and the trophozoite stages or forms. The cyst is both the infective and diagnostic stage or form, while the trophozoite is the actively feeding stage of the parasite, so it's the vegetative stage. It is also diagnostic if we get it in a fresh stool sample. The life cycle is described as direct. You know what that means. Only one organism is involved in the life cycle. Now, infection occurs when a susceptible host ingests the cyst. The cysts are described as hardy because they have a covering that is resistant to environmental conditions. So it's resistant to drying, it's resistant to temperature. So the organism can persist in the environment for some months. Now, following the ingestion of this cyst, the gastric juice, which is acidic, will soften the cystic coating. So by the time the content passes into the small intestine, where the pH is no longer acidic, the the process of excistation occurs. That means that you have trophozoites emerging from the ingested cyst. Each cyst produces two trophozoites, and immediately they are liberated, they attach to the brush border of the enterocyte, and different mechanisms have been proposed to explain this attachment. These include the fact that the organism has a disc with contractile proteins with which it latches on to the brush border surface of the enterocyte. Because it's motile by means of flagella, there is some degree of flagella-mediated hydrodynamic forces that tend to enhance the attachment to the brush border. Then there's also a receptor-ligand interaction mediated by lectin protein present on the organism. So once attached, the trophozoites will start to metabolize and then they multiply by longitudinal binary fission. They remain in the lumen of the proximal bowel. This is very important to note. The organism is non-invasive. As a result, they don't cause dysentery. So, like I just mentioned, the trophozoites are not toxigenic. They are not invasive, and neither are they toxigenic. So they don't produce toxins. What they do is a physical disruption in the brush border integrity, leading to microvilli injury, eventually causing atrophy, which now results in watery dry because the absorptive function of the intestine is compromised. How does the patient present? About a third to like half of patients who are infected are asymptomatic. But when symptoms occur, it could be a spectrum of mild diarrhea to severe malabsorption syndrome. So patients may present with fat malabsorption, diarrhea, epigastric pain, and flatulence. With chronic diseases, with multiple relapses, this has been attributed to antigenic variation associated with the parasite. How do you make a diagnosis? Stool specimen, in this case, will be a good sample to send to the lab. But note that you need to send in more than one sample because the cyst and the trophozoites are released intermittently into the stool sample. The characteristic trophozoite looks like a pear-shaped structure with pairs of flagella. The cyst has the appearance of an old man's face. Okay, so this is typical of Giardia lamblia. Other samples that may assist in diagnosis include a duodenal aspirate, a biopsy of the upper small intestine will be sent to the histology lab, not the micro lab, and under H&E stains, you can actually see the organisms in close association with the brush border surface, the intestine. Other approaches to diagnosis include fecal antigen detection using different assays like immunoelectrophoresis, enzyme immunoassays, or immunochromatography. These are all different approaches trying to detect the fecal antigen in stool samples. You can also perform nucleic acid amplification tests, either through DNA probe assays or polymerase chain reaction. Prevention, since this is fecal-orally transmitted, if there is adequate disposal of waste material, then the environment will not be contaminated by the infective form. So adequate provision of waste, human waste management, good hand washing practice, okay, washing your hands before eating food, washing your hands after using the bathroom, washing your hands after changing a baby's diaper, washing your hands before preparing food. These are all geared at physically removing the cyst on your hands if they are contaminated. We can also wash our vegetables water, particularly the vegetables that we're going to consume raw, because we are not going to cook them. We should make sure that they're thoroughly cleaned with potable water. We can also boil our water or filter it to remove the cyst or destroy the cyst, depending on which technique you're using. Another protozoan parasite of importance in the pathology in the GI tract is Entamoeba histolytica. Entamoeba moves with the aid of pseudopods. It has a worldwide distribution, but it's relatively prevalent in tropics and subtropics, particularly in areas where you have poor social infrastructures. That's why it's endemic in developing countries. About 35 to 50 million people are infected per year, and over 100,000 deaths every year is attributable to this parasite. Humans are the reservoir. This is in contrast to Giardia lamblia, where you can have animals serving as reservoirs. One thing you should note is that non-pathogenic species exist, and you have to be very careful because at times they may introduce a bias when you are assessing a patient sample. So you have to keep this in mind, and you have to look for different characteristics that can distinguish the pathogenic form from the non-pathogenic form. Just like Giardia lamblia, Entamoeba histolytica exists in two forms. You have the trophozoite. Again, this is the actively dividing and metabolizing form. So we say it's the vegetative stage. It's also diagnostic if you find it in the stool sample of patients. You also have the cyst, which is both infective and diagnostic. Infection is fecal-orally transmitted when matured cysts are ingested, either in contaminated food or water. Contaminated fruits and vegetables may also transmit the infection. You can also have transmission through oral anal sex. And apart from causing a GI GI tract infection in this situation. There are instances in which in homosexual males, there are cutaneous lesions attributable to the parasite. So we can also have cutaneous amoebiasis. We can also have vector transmissions where you have domestic flies physically perching on stools in the environment and picking up these infective cysts on their abdomen, which is hairy, and then flying into the kitchen, perching on cooked food that were not covered. So they can deposit the cyst. So although you washed your hands and you cooked the food very well, you didn't cover it after preparation. Flies perched on it. Unfortunately, these flies have visited stools in the environment and picked up the infected form, and now they are depositing it on the food. The life cycle is direct because you have only one human host involved. The trophozoites are released from the cyst in the duodenum, and these trophozoites at times could be found in the crypt of the large intestine. So this organism behaves like it's anaerobic, okay? Because at the in the crypt, the oxygen tension is relatively low. These trophozoites produce extensive nuchal necrosis in the large intestine. That is why with time, the diarrhea associated with entamoeba histolytica may become bloody. The basis for this tissue destruction is not completely known, but it's been attributed to the production of a cytotoxin. The release of toxic neutrophil content as well is believed to contribute to tissue destruction. The characteristic histological lesions associated with intestinal amoebiasis are referred to as the flask-shaped ulceration or flask-shaped ulcers. And when biopsies are taken, you can actually demonstrate these lesions. There could be invasion into deeper mucosal layers with extension into the peritoneal cavity. This can lead to secondary involvement of other organs, such as the liver, the lungs, the brain, and the heart. So this is extraintestinal amoebiasis, and it is attributable to migrating trophozoites. So look at this diagram here. What this diagram is trying to explain is that when an individual is exposed to the matured cyst and you have the infection established, in a significant number of patients, that like 90% of the cases, it's a self-limiting asymptomatic infection. So the patient hardly comes to the clinic complaining about anything. However, in about 10% of patients who have been exposed, you can have invasive disease. With invasive disease, you can have, the trophozoite can have access to extraintestinal sites, primarily the liver. But you can also have involvement of the lungs and the brain. So you can have amoebic liver abscess, lung abscess, or brain abscess. How do you make a diagnosis? With amoebic infection, stool microscopy can help. Again, you need to send in multiple samples. We look out for the characteristic trophozoites. Now, the trophozoites of the pathogenic amoeba species are quite different from the non-pathogenic ones in that they ingest red blood cells. So the presence of red blood cells within the trophozoite is highly suggestive of a pathogenic strain. The non-pathogenic ones feed on bacteria in the gut, so they don't feed on red cells. Another feature that you can look out for, or another form of the parasite that you can look out for, is the cyst. Now here you have a cyst that is not matured because you have only one nucleus. The matured cyst of Entamoeba histolytica contains 4 nuclei. The non-pathogenic strains usually have more than 4, up to 8. The caveat here is that with Entamoeba dispar, the matured cyst also contains 4 nuclei. However, Entamoeba dispar is non-pathogenic. So at times, when you have cysts showing 4 nuclei, you are not sure whether you are dealing with Entamoeba histolytica or dispar. So normally when results are sent from the lab, we report the two, cyst of Entamoeba histolytica / Entamoeba dispar seen. It is now left to the managing team to interpret the result vis-a-vis the clinical presentation of the patient. If the patient is symptomatic, the patient is having bloody stools, then you're likely dealing with Entamoeba histolytica. However, if the patient is asymptomatic, you are not sure whether this is a carrier or if this is a non-pathogenic situation. So you need to go a step further and perform other tests, such as nucleic acid amplification tests, looking out for specific genetic material associated with E. histolytica. Now, because you can also have extraintestinal infection, there are other assays such as the polymerase chain reaction, the DNA probes that you can use to make a diagnosis. Enzyme immunoassays can also be applied to the fecal sample to screen for the fecal antigen peculiar to the parasite. Again, prevention is similar to what you have for Giardia lamblia. Adequate food hygiene, hand hygiene, and water hygiene. So now we have discussed 2 conditions that could lead to dysentery. In the previous lectures, we've talked about Shigella, right? And enterohemorrhagic E. coli. These are bacterial agents of dysentery because they can cause bloody diarrhea, bloody mucus diarrhea. Amoebic dysentery can also lead to amoebic diarrhea, dysentery. So clinically, how can you make a difference? How can you differentiate between the 2 conditions? Now, in terms of the infective agents, you know that the amoeba is a protozoan parasite, whereas with bacillary dysentery, they are usually gram-negative bacteria. Escherichia coli is gram-negative, Shigella is gram-negative, they're gram-negative rods. In terms of how the patient presents, amoebic dysentery, although the patient is passing out bloody stool, but the patient will come to the clinic to complain that I'm worried, I can see blood in my stool. However, a patient with bacillary dysentery has some degree of toxemia, so they are usually brought in by close relatives or friends because they can't come to the clinic themselves. So this idea of walking dysentery and lying dysentery, they come in on a stretcher. The epidemiology, amoebic dysentery is endemic in developing countries. Bacillary dysentery may be epidemic. You may have mini outbreaks. The presence of tenesmus is usually associated with bacillary dysentery. When you send the stool to the lab, with amoebic dysentery, we tend to see reduced number of polymorphonuclear cells. And we can see trophozoites or cysts depending on if you send in a fresh sample, stool sample or not. With bacillary dysentery, however, you have a lot of inflammatory cells, polymorphonuclear cells, and then you don't see trophozoites and cysts. So this can give you an idea of what you're dealing with. Now we are moving on to parasitic agents that are regarded as opportunistic enteric parasites. You already know what opportunistic means. There's something that makes that particular host unduly susceptible. So opportunistic enteric parasitic infections are encountered in about 30 to 60% of HIV seropositive patients in developed countries and in about 90% of patients in developing countries. Now, the major predisposing factor to opportunistic enteric parasites is the CD4 count. When this count falls below 200 cells per microliter, patients are more susceptible to opportunistic infections, including enteric parasites. So those listed agents are usually associated with opportunistic parasitic infection in immunocompromised patients. However, you should note that the usual organisms such as Giardia, Entamoeba, Strongyloides can still cause infection in this group of patients. It's just that now the presentation may be atypical because of their immunosuppressed or compromised state. Cryptosporidium parvum will be the first agent we're going to discuss. Yeah, this sentence is wrong. I wanted to type in the first case in a homosexual man was diagnosed in New York in August of 1981. So it's not the first case was diagnosed, no. Human cases associated with human infections have been noted as early as the 1900s, but usually in young children. It's a leading agent of diarrhea in young children. The first case in a homosexual man with AIDS was diagnosed in August of 1981. So it became like an opportunistic agent associated with people with HIV/AIDS. There are two species that are usually implicated in human infections: Cryptosporidium parvum and Cryptosporidium hominis. Cryptosporidium parvum was formerly referred to as Cryptosporidium parvum 2, and Cryptosporidium hominis was initially referred to as Cryptosporidium parvum 1. While Cryptosporidium parvum is zoonotic because you can have animal to human transmission, Cryptosporidium hominis is associated with humans only. It is non-zoonotic. This particular agent is a coccidian parasite. It's also a member of the Apicomplexan. If you remember from the POD lecture when we were discussing malaria parasite is another example of a coccidian parasite, and it's also an Apicomplexan. It's a coccidian parasite because of its complex life cycle, which we're going to discuss in a moment. Infection is transmitted through drinking of contaminated water. Now, this water could be drinking water, even after treatment. So we are going to discuss appropriate water treatment that can prevent transmission. Or it could be through swallowing of recreational water. For example, you're in the swimming pool, or you didn't know that somebody is shedding the infective form of this agent. And during swimming, it's quite possible for you to swallow, taking some water while swimming. So infection could be acquired through that way. You can also have a zoonotic spread. So if you're a vet doctor, if you're a livestock farmer, and you're handling animal products, you're treating sick animals, this agent could be transmitted. You can also have fecal oral spread with homosexuals, people who practice oral anal sex. So what is the life cycle like? Again, this is a direct life cycle because it's just one human host. However, it is a complex life cycle because, as we're going to discuss now, you have an alternation of asexual reproduction with sexual reproduction. If you recall from your malaria parasite lecture, we mentioned it that a complex life cycle means that you have asexual generation, reproduction, followed by sexual reproduction, then another asexual reproduction, and then it goes on like that. Unlike the malaria parasite, there is no insect vector involved in the transmission of this agent. The life cycle begins when you have an infected person passing out thick-walled oocysts. These oocysts contain 4 sporozoites. Okay? These oocysts can contaminate water, can find its way onto our food, and then we end up ingesting the cyst. Sorry, oocyst is different from a cyst. A cyst results from asexual reproduction, and oocyst results from sexual reproduction. So this thick-walled oocyst is ingested, and then what happens in the intestine? After passing through the stomach, gastric acid is going to weaken that thick wall. So by the time the oocysts are getting into the GI, the cyst wall is practically gone, and you have the sporocyst emerging from the oocyst. Usually, sporulated oocyst gives rise to 4 sporozoites. These sporozoites are going to invade. Well, here, some authors differ. Some scientists believe that the sporozoite does not actually invade the enterocyte, but rather they have a close association with the microvilli, and that the microvilli will extend and change its form to encompass them completely to become a trophozoite. While others believe that the organism actually invades the enterocyte, but it stays close to the luminal surface. So when you look at it under the light microscope, you will think that the sporozoites that eventually form the trophozoites are extracellular. But whatever the case might be, there is a close interaction between the sporozoite and the microvilli in the GI. With this, there is a morphological change that occurs with the sporozoite, and it is transformed into a trophozoite. Okay? With the transformation to a trophozoite, you have asexual reproduction taking place, and the trophozoite transforms into a type 1 meront. A type 1 meront results from asexual reproduction of the trophozoite. Is that clear? When the meront matures, it eventually breaks down to liberate merozoites. These merozoites can go ahead to establish another cycle, just like the sporozoites that were ingested, and then you have continual infection of the enterocytes. However, due to some yet to be described symptoms, some of the merozoites that are released from the type 1 merons will change morphologically and become type 2. The type 2 merons will divide and release about 4 to 8 merozoites. These are type 2 merocytes, different from the ones released by type 1 merons. These merocytes that emerge from the type 2 merons will undergo some form of morphological changes and differentiate into what we call the microgametes and the macrogametes. Okay? The microgamete will go ahead to change its form and eventually break down to release microgametes. These microgametes will fertilize the macrogametes. So this is where sexual reproduction occurs, and then a zygote is formed. Okay? So here we have asexual reproduction. Here we have sexual reproduction. Now, the zygote that is eventually formed when the microgamete fertilizes the macrogamete can have either of two outcomes. One is that it forms a thin-walled oocyst. Okay, so this zygote will develop further to form a thin-walled oocyst. The thin-walled oocysts are never passed out in the stool. They eventually mature and release the sporozoites, so you can have autoinfection taking place. Another outcome of the fertilized zygote is that the oocyst develops a thick wall, and then it's passed out in the feces, and the life cycle continues. Okay? Do you have any question? If you have any question, let me know. It's a little bit confusing, I know, but if you still recall the life cycle of malaria parasite, you shouldn't have too much issues understanding this life cycle. Remember, it's complex because asexual alternates with sexual before you can have complete maturation of the parasite. But unlike the malaria parasite, it's all occurring in one host. So we don't have an insect vector that would come to inject the sporozoites. Sporozoites are actually ingested, and then they develop through the type 1 merons to the type 2 merons, and eventually the zygote is formed, which can give rise to thin or thick-walled oocysts. So you can have autoinfection, or you can have infection perpetrated in the environment. Cryptosporidium organisms are found just within the brush border of the intestinal epithelium. It attaches to the surface of the cells and replicate, leading to production of new infectious oocysts. So the different processes I mentioned in the last slides have names. Merogony refers to the process of forming the merons. Gametogony leads to the formation of the zygote. And sporogony is what happens when the zygote eventually gives rise to the oocyst that contains the four sporozoites. After sporogony, the matured oocyst may either exist within the digestive tract of the host, like we've said, that you can have the thin-walled oocyst. So that leads to infection, autoinfection of new cells, or it may be excreted into the environment. How does the patient present? Remember that the brush border has some enzyme functions, digestion of complex carbohydrates and things like that, as well as some degree of absorptive function. So when you have a functional impairment due to the fact that this organism is replicating at this site, you have a form of secretory diarrhea as well as some degree of osmotic diarrhea. But because the organism is not actively invasive, you don't have dysentery. So the stools are non-bloody. There may be slight fever, fatigue, and myalgia. The oocysts have occasionally been shown to infect the lungs, So the patient may present with chest symptoms. Because of the high amount of fluid lost per day, which can be in the range of 5 to 10 liters, patients may present in a state of dehydration. At times, you can have chronic infection because you can see the interplay of onto infection and how the merons one can give rise to merozoites that may actually go back to infect more enterocytes. So you can have a chronic diarrhea. Patient may present with extreme weight loss. So these are ways by which the patient may present. How can you make a diagnosis? The oocysts that we discussed that are the infective form are very tiny structures, about 5 microns or below, at times 3 to 5 microns. So these are very tiny structures. Under the light microscope, they are not refractile. So we need to modify the staining technique so that they can be visualized under the light microscope. So we can perform the Keon staining, which is a type of modified acid fast stain. You still remember Zell-Nelson staining, okay? That is an example of an acid fast staining technique. So this can be adapted to make the oocyst visible. And here you have a micrograph showing this. Immunofluorescence staining can also be used. We can raise monoclonal antibodies against antigens associated with the oocyst wall. So you can actually put antibodies onto fluorescent compounds and then you look at it under the fluorescent microscope. There are also lateral flow immunoassays like the one shown here, where you have a negative reaction and a positive reaction. Then we can also use hematochromatographic assays as well as molecular assays. But the molecular assays are not, routine assays. You may use them in research institutes and for research purposes. Prevention can be achieved by proper treatment of water, drinking water. And we are going to discuss what constitutes proper treatment of water. Safe disposal of human waste can break the chain of transmission because the oocysts we know are the infective form. So if it's not present, if it's not contaminating the environment, the risk of transmission drops. Good hand hygiene is also very important. After using the bathroom, wash your hands so you don't introduce the oocyst onto food or transmit it to close friends. So this is important for us to know, particularly when we travel to endemic areas. We have that notion that it's bottled water, it's safe. Not all bottled waters are created equal. That's why I like this slide. So you have to pay attention to how the water was processed. Water so labeled has been processed by these methods: reverse osmosis treated, distilled, microfiltered, filtered through an absolute micron or smaller filter. These are effective against crypto. Remember, I told you that the oocyst is about 3 to 5 microns. So if you use a filter that is about 1 micron, you are likely to get the oocyst out physically. Distilling the water ensures that what you have is the condensate. Okay? But if the water has the label carbon filtered, the pore size is too large to to filter out crypto. Okay? If it says filtered, it didn't tell you the size of the filter, it's probably not effective because the regular filter will not filter out the oocyst of crypto. If it says particle filtered, multimedia filter, ozonated, ozone treated, ultraviolet light treated, all these are not effective against crypto because the oocysts are highly resistant. Okay, even chlorine is not enough to to kill the oocysts in the environment or in water. Now, we move on to another parasitic agent. This is Strongyloides decoralis. I think this is the first time I'm going to discuss this with you. During POD, we only discussed hookworm. Strongyloides is another nematode that may be confused with hookworm if we are not skilled enough to know the difference between the larval form. Okay, so if you get the larva of Strongyloides, it's so similar to that of hookworm. However, they are not the same. They have distinct characteristics. So Strongyloides pecoralis is a nematode that is telling you that this is a roundworm. So being a roundworm, if you recall from your POD lectures, it means that it has a complete digestive tract. So there's a mouth part and there's an anus, and this organism can actually feed. Okay. However, unlike hookworm, the eggs of this particular agent hatches in the intestine, so they are never passed out in the stool. So we don't get to see the eggs of Strongyloides decoralis in stool samples of infected patients. Excuse me. Another difference is that the larvae, when they emerge from the eggs, they can mature into the filariform. The third stage larvae are the filariform larvae, and they are the infective form. They are the ones that can actually, penetrate through intact skin or penetrate through the wall of the GI. So you can have autoinfection, which is not common with hookworm, because with hookworm, the eggs are laid, the eggs will embryonate before becoming infective. This parasite, just like hookworm, may undergo a free-living non-parasitic cycle outside the human host. So the female worms lay the eggs in the gut wall, like I said earlier, and there are two forms of the larvae that we can see in stool samples. The rabidiform larva, or the second stage larva, is the non-infective form. The filariform is the infective form. Now, there are ways by which we can differentiate between the two. I'll mention them, but I don't think it's important for you to know. We can look at the esophagus of these worms, and we can look at what we call the esophagus bulb to make a difference between the stage of development, and also to speciate, to differentiate them from the larval forms of hookworm. You don't need to know this. I just want you to know that there are things that we can use in the laboratory when we get these juvenile worms that can help in diagnosis. Now, infection can be acquired through skin penetration, wading through contaminated water, just like with hookworm, just like with schistosomiasis. The larval form can actually, and we can also have autoinfection when these eggs release the larva in the host, and then the larva matures to the third stage form, which can establish the infection. So let's look at what the life cycle looks like. Now we're going to consider the life cycle in humans and what happens in the external environment. So far, studies seem to suggest that the female worm is located in the submucosal surface of the GI, and that's why infection with strongyloides is associated with a high degree of eosinophilia. It is a fact that with most helminthic infections, there is a degree of eosinophilia, but because of the location of this worm, it is believed that the degree of eosinophilia is higher relatively compared to those of other helminthic agents. But be that as it may, the female worm is known to produce parthenogenesis. That means that this female worm lays her eggs without mating with a male worm, and the eggs develop embryo not too long after being passed out. So that's a peculiarity of this particular agent. So when the female produces eggs in the mucosa, remember we said that the eggs will embryonate, and then before you know it, the juvenile worm will emerge. The juvenile worm will develop to the filariform, which is the infective form. And this can, depending on where the development took place, it can either penetrate the intestinal wall or perianal skin. It gets into the circulation. You still remember that we have this heart-lung migration, particularly with roundworms. And then eventually it gets to the trachea, it's swallowed, and then the adult worm develops. So this is the usual thing. So this is a direct life cycle. However, at times, the juvenile worms are passed out in the stool. In the environment, they can actually develop into separate sexes. So you have the female and the male worm, but they are feeding on environmental microbes. They are not causing any infection. They can mate and they start producing eggs in the soil. Usually the soil must be warm and moist. That's why these agents are prevalent in the tropics and the subtropics. Then you have indirect development. The juvenile worm emerges again from the eggs. It eventually matures to the filariform form in the environment. And then when somebody wades through contaminated environment, the life cycle starts all over again. There's penetration of the skin. It gets into the circulation to the lungs, the trachea, it is swallowed, and then the adult one in the GI again. So this is the peculiarity of Strongyloides decoralis. How does this organism, infection with this organism present? Strongyloidiasis, which is the condition that results when an individual is infected with Strongyloides decoralis, can present in three ways: the acute, the chronic uncomplicated, and the disseminated form. Now, out of these three, the disseminated form is the most lethal because it tends to occur in immune compromised patients, and you can have the worms in any site of the body. Remember, I told you this is a round worm. They can actually eat their way through various organs. So they can cross into the brain, you can find them in the lungs, you can find them in the heart, anywhere they feel like going, because they are distributed widely. With acute strongyloidiasis, it's usually seen in children, it's manifested by diarrhea. There may be associated low plasma protein and low serum potassium. With the chronic uncomplicated strongyloidiasis, patient may complain of recurrent epigastric pain, recurrent hives, transient red lines on the skin as shown in the micrograph, and episodic diarrhea. The disseminated strongyloidiasis is also referred to as hyperinfective syndrome or hyperinfection. The range of manifestation is quite diverse. Patient may present with sepsis because this organism resides initially in the GI where you have a normal microbiota. By the time it eats its way out and gets into the blood circulation, it is coated by the microbes, the normal microbiota in the gut. Remember that when the microbiota are in the gut, they don't cause any trouble. But when they get to a sterile site, like the bloodstream, then it becomes a problem. So this organism serves like a vehicle that moves organisms from the gut into the bloodstream. So the patient may present with sepsis. There could be respiratory symptoms. You could actually have gut signs like perforation, intestinal perforation, and things like that, because the worms actually ate through the gut wall. So you may also have peritonitis and things like that. Because the patients that usually uh, present with disseminated infection are immune compromised, there is a high degree of case fatality rate. Um, so immunocompromised patients, patients who are immunosuppressed, maybe because they received an organ transplant, or malnourished children are at particular risk of developing hyperinfection. However, there have been some case reports that does not have any of these predisposing conditions, and they still came down with a hyperinfection. So maybe at times you have some form of interplay between the parasite and the host. There could be other manifestations of strongyloides speckularis infection apart from what we've just mentioned. So the patient may present with paralytic eyelids, patient may present with severe diarrhea, there could be pneumonia, there could be pulmonary hemorrhage, there could be septicemia, you can have other bacterial infections super added onto it. How can you make a diagnosis? Feces can be sent to the lab to look out for the larvae. But the problem here is that this is not a very good sample to send to the lab because the worms are released intermittently. You may need to send up to 8 samples to the lab before the lab can say whether the patient is infected or not. So this is not the best approach to make a diagnosis. Microscopy using stool sample is not the best. Serology may help. ELISA to detect the antigen in blood. And then you can do what we call the culture approach. And one of the good ones is the Harada-Mori filter paper method. Although we say it's culture, but it's misnomer. This is a personal opinion because you're not actually culturing anything. What you're doing is you're taking the patient's stool sample and you're going to smear it on to a filter paper, a strip of filter paper, and you're going to immerse that filter paper in warm distilled water. And you make sure that you have at least an inch of interface between the water and the stool sample. So the water should not actually touch the area where you have smeared this patient's stool on. And then you're going to incubate this at room temperature, which can range from between 25 to 30 degrees centigrade for 10 days. And every day, what you do is you're going to take a small aliquot of this, at it under the microscope. Anytime you take out of the water, you must replace it because it mustn't get dry. Now, the reason why this is done is because those larvae that we discussed, the rapidiform, they have a predilection for water. So keeping the tube relatively moist will draw the larvae from the sample to the water that you have provided. So each day for the next 10 days, you're going to be taking drops from that water and you're going to be looking at it under the microscope and looking out for structures like this. Okay. We also have the filter paper slant culture, which is similar to the Harada-Mori filter paper. This way you can make a diagnosis and looking out for the larvae under microscopy. How can you prevent infection? Infection measures should be enforced in an index patient with hyperinfection syndrome. But remember, we said the hyperinfection syndrome is associated most of the time with people who are immune compromised. So you're likely going to admit them, and you have to show strict contact precaution. You have to isolate these patients or you perform cohort nursing. Okay. Health education is also important. People in the environment, wearing of shoes will prevent the infected form from having access to the skin. Then proper sanitation and waste disposal of human waste is very important. So provision of uh, uh, water closets and what have you, pipe-on water for hand washing, all these are geared at preventing infection. Any questions? In the chat box, any questions?
No, doctor, there are no questions in the chat box.
Okay, so thank you so much. Tomorrow I'll have my last lecture with you and we'll be discussing viruses. So enjoy the rest of the day. Bye.